Today we're going to be talking about cell communication. Our first part is a G protein pathway. The G protein pathway is the most used pathway by cells. It begins with a G protein receptor that acts as an attachment point for signaling molecules. A G protein, held inactive when bound with GDP, acts as a relay protein carrying messages to the correct location, such as an enzyme. When a signaling molecule binds to the receptor changes shape and binds a G protein. This shape change causes phosphorylation to occur, causing a GTP to displace GDP. This activates the G protein. The newly activated G protein then dissociates from the receptor and binds to an enzyme. Now activated, this enzyme can trigger the next step in a cellular response. The next part is a tyrosine kinase pathway. Before the ligand binds, receptor tyrosine kinase proteins exist as individual monomers. The binding of signal molecules instigates a process called dimerization, where two receptor monomers associate closely and form a complex known as a dimer. Each dimer has 6 tyrosine kinase regions, which allows it to activate 6 relay proteins at one time. Dimerization activates each region of the dimer, causing each region to add a phosphate from an ATP molecule. Specific relay proteins recognize the now fully active receptor and bind to a phosphorylated tyrosine. The relay proteins are activated and open transduction pathways which trigger cellular responses. Tyrosine kinase pathways can act like a catalyst by activating 6 relay proteins at once. This is useful for cell growth and repair. The next part is the ion channel receptors. Also known as ligand-gated ion channels, these membrane receptors can act as a gate. The gate stays closed until a ligand binds with the receptor. Once the ligand binds with the receptor, the gate opens and allows specific ions to flow into the cell, such as sodium and calmodulin. When the ligand dissociates from the receptor, the gate closes and stops the flow of ions into the cell. These receptor proteins act as a control for cell signals. The final part in cell communication is intracellular receptors. These are mainly used for receiving hormones and steroids. Due to the fact these molecules are lipids, they diffuse into the cell across the phospholipid bilayer. As an example, a hormone diffuses across the membrane and binds to the receptor protein. This receptor complex then enters the nucleus and binds to specific genes. The bound protein stimulates the transcription of the gene to mRNA by acting as a transcription factor. The mRNA is then translated into a new protein. Cells also use secondary messengers to communicate. One example of this is IP3, or inositol triphosphate. It begins in the G protein pathway. When a ligand binds with a receptor protein, the G protein is activated. This in turn activates the enzyme phospholipase C. Phospholipase C, now activated, breaks down a phospholipid into IP3 and DAG. The IP3 then diffuses across the cytosol to the endoplasmic reticulum and binds with an IP3 gated calcium channel.
This opens the channel, allowing calcium ions to flow into the cell to help with other cellular activities, such as opening transduction pathways.
That's it for cell communication.